Good evening. I'm inferring from the fact that the lights went down that we're supposed to begin. My name is Andy Knoll. I'm a professor of biology and earth sciences here at Harvard, and it's my great pleasure to introduce our speaker tonight, Jack Szostak. Before doing that, let me just point out that this is actually the first of 4 lectures in a spring series called Evolution Matters. Later this month, Ian Kuzan from Princeton will give a talk on behavior. And there's more information on this series at the table at the right, and I would certainly encourage you, if you don't know the whole schedule, to pick up a copy and join us later on in the semester. I'd also like to acknowledge Dr. Herman Suit and Dr. Joan Suit, who are here this evening, who have generously provided sponsorship for this program, which is greatly appreciated. Yes, absolutely. I would also point out that tonight's talk is being videotaped and will soon be posted for viewing on the museum's website, so those of you who were conflicted about missing the beginning of the Celtics game now have a solution. To our speaker, Jack Szostak grew up for the most part in Canada, went to McGill University, then got a PhD at Cornell, and after some time as a postdoctoral researcher joined the Harvard Medical School in 1979. Today he is professor of genetics at the medical school. He is the Alex Rich Distinguished Investigator at the Massachusetts General Hospital, and he is an investigator of the Howard Hughes Medical Institution. Jack has had several phases of his career. The first phase, in some ways, involves some pioneering studies of telomeres, which are the molecular caps at the end of chromosomes, and telomerase, which is a remarkable enzyme that actually maintains that cap through repeated rounds of cell division. Understanding these has proven to be a key in everything from aging to cancer, and that work actually went pretty well, at least by evidence of the 2009 Nobel Prize in Physiology and Medicine, of which Jack was a co-recipient. But that's actually not why we're here tonight. For much of the last 20 years, Jack and his lab have been doing equally pioneering research on what must be the most fundamental and difficult of all biological questions, and that is, how did life originate? At the time that Jack entered this particular area of research, the research was dominated by synthetic organic chemists, people who would take what we might call a bottom-up approach. How do we go from simple inorganic precursors to the molecules of life? And Jack, along with his friend and colleague Jerry Joyce on the West Coast and others, was really instrumental in saying, can we look downward from the complexities of the molecular composition of cells that we see today and try and understand from a top-down approach how that molecular complexity may have arisen. And this involved some work that we'll probably hear a little bit about tonight on how natural selection may have been important not only after life began, but in the origin of life itself. There, of course, is a meeting ground. It's the great set of questions of where that top-down and bottom-up approach to the origin of life meet. And over the last several years, Jack's lab, again, has been leading in trying to understand the key issues of how we actually go from the synthesis of molecules on the early Earth to something that can replicate, evolve, be enclosed in a compartment. And so that's what we're going to hear about tonight. His title is The Origins of Cellular Life. Jack Szostak.
Okay, thank you very much, Andy. So one of the things that makes studying the origin of life so much fun is that it encompasses such a broad sweep of problems. Everything from planet formation the early evolution of planetary atmospheres, all the chemistry that goes on a young planet, the gradual buildup of more complicated molecules, the aspect that we've been focusing on, which is how, once you've made those molecules, how they get together and start acting like primitive cells. And then, of course, once you've reached that stage, there are all the interesting ways in which Darwinian evolution comes into play and allows organisms to adapt to their environment and become increasingly complex and interesting. So rather than sort of give a totally superficial overview of the whole sweep of the problem, what I'd like to do is try to give some background and then tell a few stories that illustrate the way that time and again apparently really difficult problems have arisen and have been really roadblocks to progress for sometimes quite a long time. And then show you how the solutions to those problems have come about, often just completely from left field, in very surprising ways. And then after that, of course, everything looks simple and obvious. So problems that look totally intractable, once we understand them, tend to, in retrospect, look incredibly simple, and we can't believe we were so stupid as to not be able to figure them out earlier. Okay, so let's see. So one of the things that has brought this field, I think, back into broader awareness is the discovery of so many extrasolar planets, right, by many astronomers using a wide range of different methods. So, if you're lucky enough to live in a place far enough away from the city lights so that you can actually see the Milky Way, you look up at it, you can know now that there are most likely hundreds of millions of Earth-like planets orbiting sun-like stars in orbits similar to that of our planet. So there are certainly many places out there where there are planetary environments that could sustain life. Okay. But what we don't know, even now, is whether, of course, there actually is life in any of those numerous other environments. And really, this all comes down to the question at the top here. Is it easy for life to emerge from the chemistry of a young planet? Or is that something that's really hard because there's some bottleneck, some particular stage in the chemistry, for example, that is just so difficult to get over that the emergence of life might be an incredibly rare event. In fact, maybe this is it here on our planet in the extreme. So it's going to be a while before we can get any observational evidence of whether there's life on these other planets. And so that is what I think has inspired me and a lot of other people to try to go into the lab and do some simple experiments and see if we can get some clues as we try to work out a plausible series of events that go from really simple chemistry to complicated chemistry to simple biology up to complicated biology. If we can show that all the steps along a reasonable pathway look straightforward, then I think it would be reasonable to suspect that life is abundant out there in the universe. But if we can identify particular aspects that are hard, then we might come to the opposite conclusion. So I have to warn you, at this stage in our thinking, there are a lot of gaps in our understanding. Right? There are many places in that chain of events where we really have no idea what happened. And so at the moment, a lot of those steps look hard. Right? But I want to show you a few examples of steps that in the recent past looked hard and now look easy. So I think the lesson is that we have to just keep exploring and doing experiments and trying to work out a plausible pathway. Okay. All right. So one of the things that held back progress in this field for really a long time, for decades, is the fact that modern life is incredibly adaptable, right? Our planet is just heavily colonized. This is one of those iconic images of a deep sea hydrothermal vent, and you can just see there's just abundant life in this really extreme environment. And many other extreme environments where, 20 years ago we never thought life could exist, and yet now we know in very acidic environments, very cold environments, very alkaline environments, there is life. Okay. But what this is telling us is something about the power of Darwinian evolution that allows organisms to adapt to these environments. What it's not telling us anything about is how easy or hard it is for life to actually get started. Okay. And so the problem is that this highly adaptable life that we're all so familiar with is also incredibly complicated. Right? So this is just a picture of a eukaryotic cell showing the cytoskeleton, all kinds of different filaments that are organized to separate the chromosomes of the cell during cell division. So very complicated structures. If you look sort of underneath that, at the next level down, cells have incredibly complicated chemistry, right? Anybody who's ever had to learn metabolic pathways in college will appreciate that. So this chemical complexity it's really hard to understand how this could have arisen in the first place. Right? And so people spend a lot of time trying to think, and they still do, trying to think about, are there primitive kinds of simple metabolic pathways that might have existed on the early planet and become incorporated into what modern cells use. But actually, if you look at the level even underneath this, the problem is even worse, because the fundamental organizational structure of all life in terms of the flow of information is also really complicated. And it's complicated in the sense that all the different aspects of it depend on each other. So we have down the middle the central dogma, right? Information stored in DNA, transmitted to RNA, which is made by enzymes. There's the RNA both codes for and makes proteins which carry out the functions of the cell, most of the biochemistry, the chemical reactions that you saw in those huge metabolic networks, all the structures of the cell. But to do this, right, it takes proteins to make DNA, it takes proteins to make RNA. RNA is involved in its own processing. You actually need RNA also to replicate DNA. So it's an incredibly self-referential system, and it was very hard for decades for people to think about how such an interdependent system could arise, because you can't begin with any one of these components. So people came up with all kinds of really Baroque, harebrained schemes by which all of these things would kind of simultaneously emerge. And the answer, when it actually came out, was very simple. And this came from experiments done by Tom Cech and Sid Altman in the early 80s. They were studying some chemical reactions that go on inside cells, trying to purify the protein enzyme that was responsible for carrying out those reactions. And they purified and purified, and what they came up with was an RNA molecule. They showed, despite widespread disbelief at the beginning, that RNA could actually catalyze chemical reactions. Okay, and that completely changed everybody's thinking about this field almost instantaneously, because now we could imagine a much earlier, simpler time in the history of life when biochemistry was basically carried out by RNA molecules. So RNA molecules that would replicate themselves, that would carry out metabolic functions, that would do things that were useful for the cell in which these RNA molecules lived. And basically, the idea was that DNA came along later as a way of archiving information in a sort of safer, more stable form. And proteins came along later because they're probably intrinsically better at doing a lot of things, at being catalysts and making structures. But as a way of getting started, this is fantastic, right? Because now all we have to worry about is how did we go from the chemistry of the young planet to making RNA and simple cells that contain replicating RNA molecules. Okay, so that has really reframed all of the issues, all the thinking about the origin of life. All right. There is, I should say, quite a bit of evidence of various kinds in favor of this, idea that there was this earlier stage of life. It's often referred to as the RNA world. The most striking and convincing piece of evidence comes from this picture, which is an image of the ribosome. The ribosome is the cellular machine that makes proteins. And so this is from a crystal structure from Tom Steitz's lab in Yale. And all the gray squiggly stuff is RNA. So most of the ribosome is actually RNA. The gold squiggly things are the proteins. They're all on the periphery. And where the actual chemistry of making proteins takes place is right here in the middle where this little green thing is. And everything around here is RNA. Can see. So it's RNA molecules which make all of the proteins in all of modern biology, including all of us. And so it just doesn't make sense to think that, for example, proteins must have come before RNA when even now it's RNA that's involved in making proteins. Right? So presumably RNA came first. Okay, so the real question is how did that happen? Okay. So here is a sort of big overview of the whole process. This is a figure taken from a review by my friend Jerry Joyce. And I really put it up to show you mostly how little we know. Okay, so some things are fairly well constrained, like the time, the age of the Earth itself. As the Earth cooled down, eventually we had liquid water, oceans, lakes, and so on. The time of this has actually been pushed back, probably closer to 4.4 billion years ago. And then sometime over the next hundreds of millions of years, there was this increasingly complex chemistry developing on the early Earth. And we don't really have good evidence, so fossil evidence for real recognizable cells until somewhere around 3.5 billion years ago. So there's a period of almost 1 billion years after the Earth started to cool down, where all of these key events must have taken place. But exactly when in there that was happening, we don't really know. There are many questions about what the environment was like that remain very controversial. And so I'm not going to get into that. I'm just actually going to focus on first of all, on some of the simple chemistry. So the problem, as I was saying, is how do we get from simple chemistry to RNA molecules? Right? So I just want to say a little bit about, just to emphasize why we need to really worry about that problem. So this is a schematic image of what we think simple cells might have looked like. They would have some kind of primitive cell membrane, basically to keep stuff inside from just floating away and diffusing into the environment. And also so that molecules inside that do something useful can give themselves an advantage. Okay? So inside, we would have some kinds of genetic molecules. Now, it could be that those were RNA molecules right from the beginning. It's still quite possible that RNA is so complicated, so hard to get to, that there was some progenitor, something simpler, maybe easier to make, maybe more stable, that came first, and when this was then taken over by RNA molecules. So RNA or something like it had to be in there to transmit information. So as its sequence gets copied and then the copies get distributed to daughter cells. Right? You can transmit information. And molecules like RNA, as we've seen because they can be catalysts, can carry out chemical functions. They can carry out metabolic reactions. They can build structures. So these are the molecules that are critical along with this cell membrane. Okay. So how do we get to these larger, more complicated structures and molecules? So these are the really simple kinds of building blocks that we're pretty sure were around. I mean, they're abundant in interstellar space. There are molecular clouds. All of these simple molecules and many more complicated molecules have been detected. You also find these things, of course, on meteorites, on comets. So I think these are reasonable, simple building blocks. And then there's a series of chemical steps that people are still working on that let you go from these simple molecules to more complex molecules. The kinds of fatty molecules, soap-like molecules that can build primitive cell membranes. The nucleotides that are the building blocks of RNA and related molecules. Amino acids that can make peptides that might help out membranes in RNA. All of these things then presumably somehow come together to make really primitive cells, protocells. Okay. So the first steps, this is, 60 years ago now, the famous Miller-Urey experiment. At first it looked like this was going to be easy. Right? So what Stanley Miller did when he was a student was to take mixtures of gases, those really simple building blocks, and subject them to an electric spark and circulate water through. And over a few days, you get this kind of really messy mixture of compounds. And in there, everyone was stunned to see amino acids, right? The building blocks of proteins. So making amino acids is really simple. And that inspired and so this is also, it's nice because this is something that you can easily imagine happening on young planets. So this is a picture of a volcano that was erupting in Chile recently. It's a time lapse, so it's just to illustrate the incredible amount of lightning that occurs in volcanic environments, where a lot of these gases are being emitted. So this kind of spark gap type chemistry is certainly, I think, is quite plausible. Okay. The question is, how do you get from that to something as complicated and delicate as RNA? So here's an image of a double helical RNA molecule. You might barely be able to trace the backbone of the structure as it winds in its helical path. Right? So it's a very complicated molecule. It's very delicate. There are definitely going to be a lot of steps going from things as simple as water, methane, ammonia, carbon monoxide, up to something as complicated as this. So let's see. So how do we think about that? So the first step really is maybe not to worry so much about the whole structure, but the repeating building blocks that make up that kind of molecule. And these are nucleotides. So these are the modern building blocks. Basically, there's a sugar part of the molecule, a nucleobase part of the molecule. This is what takes part in the base pairing in the Watson-Crick duplex. So this is for molecular recognition. This is part of the backbone. And then there's a phosphate that is also part of the backbone. Okay. So modern cells use these nucleoside triphosphates, and enzymes are very good at converting these into long chains of RNA. At the beginning of life, when we didn't have enzymes like that, we'd have to use molecules more like this. So these are more reactive, but they have the same basic structure, right? The nucleobase attached to a sugar, attached to a phosphate. Okay. So the obvious way to think about it is, okay, these building blocks have a nucleobase part, a sugar part, and a phosphate. Okay. Let's not worry about the phosphate for now. The obvious problem is, first, how do you make a sugar? Then how do you make a nucleobase? Then how do you put them together, right? If we could do that, it would be simple. Okay, so when people started thinking about this, at the beginning, it looked like it might actually be really easy. So if you look at the sugar that's part of the building blocks of RNA, that sugar is ribose. And what it is, really, is just 5 formaldehydes put together in the right way, okay? So formaldehyde is one of those really simple molecules. It's made in atmospheric chemistry. It's even made today. It's very simple to make, and so if you could concentrate this and then somehow get 5 of them to come together in the right way to make this sugar, then we'd be done. Okay. The problem is, it's actually not that simple. And when you try to polymerize formaldehyde molecules, you don't just get ribose. You get every conceivable possible sugar, and there are hundreds of them. Okay, so you only get a tiny fraction of ribose. So a lot of people have concentrated on trying to find ways of manipulating the pathway, channeling the reactivity to get more and more ribose. And there's been a fair amount of progress on that, I have to say. Okay, what about the nucleobases? At the beginning, that also looked like it might be a very simple problem. Okay, so adenine, the A of RNA, turns out, is just 5 cyanide molecules put together, right, in the right way. So you might be noticing that all the building blocks of life are incredibly poisonous, deadly compounds, which is one of the ironies of the field. But anyway, cyanide, again, very easy to make in high-energy atmospheric chemistry. Get 5 of them, put them together in the right way. You can make adenine. In fact, Juan Arrow, more than 50 years ago, just boiled some cyanide and found that, in fact, he had made some adenine. So that was an incredibly exciting result at the time. The problem is the same problem with the sugars, right? You don't just make this. You don't make what you want. You make dozens of other compounds. And again, there's been a lot of effort put into trying to channel the reactivity and get more of what we want and less of what we don't want. But the real problem, the other building blocks other than A and G are C and U, the pyrimidines. These are actually easier to make. Again, very simple building blocks. These 2 molecules will come together and make cytosine, or C. And then when this just sits around in water, you make uracil, or U. Okay, so the pyrimidines actually are not so hard to make. Again, you make a lot of other molecules, right? The right ones are not the only thing you get. But the real problem is that even if you could make the right sugar and just the right bases, you can't put them together. Right? And this stumped people for 30 years. So this is what we need biologically. This is the biological building block, one of the 4 building blocks of RNA, right? And if you just look at it, you've got this ring compound here, this ring compound here. It just seems obvious that this is the place to divide them, and you should make this part and join them. But it just doesn't work. Okay, so how is that? It turns out that that problem has been largely, if not completely, solved just within the last few years from really incredibly creative work from John Sutherland's lab in the UK. And the approach was to say, well, maybe we don't have to make the sugar and make the base and join them. Maybe there's a different way, right? And what they figured out is, in fact, you can use some of the same building blocks, but put them together in a different order. Right? So the same building blocks that would have gone into making the sugar and the base, if you let them react first, you make this weird compound here that nobody ever cared about before. And then in a series of simple steps, you can take this intermediate and build it out into the complete nucleoside. And all the steps work really efficiently. So you get mostly the right stuff at the end. So a simple problem in chemistry, right, that stumped people for decades. Turns out when you look at the problem from a different angle, there's a solution that in retrospect seems obvious. Okay. All right, so I want to switch gears now and tell you a similar story about a different aspect of the problem, about these primitive cell membranes. Okay. So let me start off by saying to make primitive cell membranes, what you need are molecules that we call amphiphiles. They have part of the molecule likes to be in an oily environment, part of it likes to be in water, and that makes them like to line up in sheets. It's pretty easy to find molecules like that in nature. You can find them in meteorites, and they'll make these beautiful little structures you see here. There's been experiments done to mimic the chemistry in interstellar dust clouds, and again, you make molecules that can spontaneously assemble into droplets and hollow vesicles. This is another experiment using high temperature, high pressure conditions that might exist sort of deep down below hydrothermal vents. Again, you can make these really beautiful structures from very simple chemistry. One of the scenarios that I like is that these kinds of molecules might have been made deep down in the earth, below hydrothermal vent systems, where you have transition metals like iron and nickel that form crystal surfaces that catalyze this kind of chemistry. So this is an animation that was made by Janet Iwasa when she was in my lab. And what you're seeing here are molecules of hydrogen and carbon monoxide sort of jiggling around over the surface and gradually self-assembling into these linear chains with oxygen atoms at the end. And then eventually they detach from the surface, and then they can get carried up to the surface by the flow of water, and in a somewhat fanciful scenario, brought to the surface in a geyser and blown over the land. Okay, so the chemistry to make these molecules is pretty straightforward. The way they actually behave in the lab, in practice, is it's really fun and beautiful. So we often use molecules like oleic acid, simple fatty acid that comes from from olive oil. If you shake this up in water with some salt and buffer, those just spontaneously assemble into beautiful structures like you see here. So they make sheets of membranes that close up into spherical particles we call vesicles. Here's another example. It just shows you how complex and messy and heterogeneous these mixtures are. So they're giant vesicles, they're little tiny vesicles, there are vesicles that have thousands of little vesicles inside them. So it's a real heterogeneous mess. So the problem, when we started thinking about primitive cells about 10 or 12 years ago, the problem we were wrestling with was, okay, these kinds of structures are really easy to make, but how can you make them grow and divide? And especially the problem of division seemed really very difficult. We were fairly easily able to show that we could make small vesicles divide in a way that was very artificial. Right? So if we take big vesicles and squirt them through tiny pores in a membrane, sure enough, you get small vesicles coming out the other side. Okay. So on the one hand, that was nice because it's a proof of principle that you could use a simple physical process to drive an analog of cell division. Right? Even though modern cell division is an incredibly complicated process that uses a huge amount of highly evolved machinery, the problem was this was not very satisfying from the point of view of thinking about cells, primitive cells on the early Earth growing and dividing. And physically, the problem is that when a cell grows as a sphere, which is, as you saw from those pictures, what these vesicles look like, they form spheres, it's really hard to make a sphere divide into two smaller spheres. It takes a lot of energy to do that. Basically, you lose some of the volume because the volume of the smaller daughter spheres is less So that's not very satisfying. And we really couldn't come up with a plausible physical process by which primitive cells could divide on the early Earth. So, you know, it really took us several years of thinking about this problem. And if we had really only focused on how to divide spherical cells, we never would have come up with the answer. In fact, the answer came from studying different ways in which primitive cells might grow. So when we started off, we were thinking of things growing as spherical cells, small spheres growing into big spheres, and that left us kind of stuck with how those spheres would divide. But eventually, after studying how these things could grow for a long time, we finally said, look, we really want to just watch how these things grow in the microscope so we can actually just see what happens. And so in a series of experiments that were done by Ting Zhu when he was a student in the lab, Ting was able to make fairly uniform populations of spherical vesicles and then just add more food, more fatty acids, and watch how they grow. And so we were expecting them to grow sort of from small spheres to bigger spheres, or maybe if the surface area grew faster than the volume, we thought that they might become a little bit elongated. So what we didn't expect is what you can see here. I hope you can see it. All the spherical vesicles have little tails growing out their sides. Right? So that was just totally bizarre. We were really shocked. This is after about 5 minutes. After about half an hour, all the spherical vesicles had turned into these long filamentous structures. It's actually really cool to watch this happen, so I'm going to show a movie of it happening. And I don't know if we can turn off these front lights because the movie's a little dim. But if there's someone who could do that, it would probably help. Is there stage lights? Oh dear. Oh, excellent. That was easy. All right, here it goes. So you can probably see a thin little filament growing here from this vesicle. Over time, this is speeded up, but over about half an hour, this filament gets longer and thicker. It gets brighter as the contents distribute throughout the length. Whoa. No, off. Yeah, we had it. It was good. We're good. Okay. Anyway, you get the idea. Spherical vesicles, if you dump in a lot of food, grow into long filaments. Okay. And that basically solved the hard problem that we'd been worrying about for almost 10 years, right? Because these filaments, unlike spheres, are incredibly fragile. And all you have to do is blow on them. A puff of air will cause them to fragment. A gentle shaking will make them divide. And there are actually other ways of making them divide. So I'm going to show you a couple of other movies because it's kind of cool to watch this actually happen. So here is something that was one of these filamentous vesicles. It forms this kind of string of beads morphology, and then they snap. A little shear force in the solution makes them snap into daughters. Okay. It's always cool when there's more than one way to make something happen. And so this is something we've been studying recently. This is another way of driving division that uses light and light-induced chemistry to cause a filamentous vesicle to break up into a whole string of beads, and then the individual beads separate and diffuse apart from each other. Okay. So once we understood this different way in which primitive cells could grow, the problem of division basically solved itself. Okay. All right. So putting those things together, we have actually a fairly robust cycle of growth and division that can actually, you can go around this cycle as long as you want until you get bored to death. Okay, it's fairly robust. I think it's something where you can actually imagine reasonable scenarios on the early Earth where something like this could actually happen. Okay, so that's very nice for the growth of a primitive cell membrane, but really it's nothing more interesting is going to happen unless you can have molecules like RNA molecules inside that are also replicating and allowing these structures to adapt to new environments, to become more complicated, to grow and divide in more efficient ways. Okay, so for that, we need to go back to nucleic acids, RNA and related molecules. Okay, so just to emphasize again that they can transmit information, but they can also do useful functions. Okay, so we need to have a way in which these molecules can replicate. And so, of course, in all modern cells, this is done by very complicated and very efficient protein enzymes. But in the beginning, there wasn't anything like that around. So we have to think of simpler ways of doing it. Shortly after the discovery of the fact that RNA molecules could catalyze reactions, everybody jumped to the conclusion that RNA could act as an enzyme to copy its own sequences. Right? So, and this is the so-called RNA replicase, an RNA molecule that's an RNA polymerase, good enough to copy its own sequence. We and others have spent a long time trying to actually make molecules that can do that. There's been a lot of progress, but we're still not there. And still, there's actually still quite a long ways to go. There was an older model, though, that maybe the chemistry of the early Earth would actually provide a pathway by which RNA or similar molecules could replicate, probably not very efficiently, but in a much simpler way. And so this is something that we've been studying for the last several years. Other labs, most notably the late Leslie Orgel, started studying this problem actually in the 60s. So right away that tells you it's not a simple problem. In fact, it's complicated for a lot of different reasons. So what I want to show you is first another one of Janet's movies, just to illustrate what we're really after. And then I'm going to briefly go through all of the reasons why this pretty picture doesn't work. Okay, so we want to start off with a strand of RNA and floating around in solution with activated building blocks. We'd like these building blocks to find their partners on the template strand by base pairing, Watson-Crick base pairing, and then just click into place using spontaneous chemical reactions. Okay, so that means we need to really think about what these building blocks are, what kind of chemistry is going to allow this to happen. And so for 40 years, people have been thinking about this problem. And on the one hand, a lot of progress has been made. On the other hand, there's a whole list of problems here that have stood in the way of being able to copy sequences in any general efficient manner. Some of the most obvious are that all the reactions we're looking at are too slow. They're on the same time scale at which RNA molecules fall apart. So that's not very satisfying. The process isn't accurate enough. The fidelity of the replication is not good enough to allow any kind of useful information to get propagated from generation to generation. There's a problem called the regiospecificity problem. Don't worry, it sounds complicated, but I'm going to explain that because this is one of these problems we've been thinking about where we think we might now finally have the answer. But for years, it was thought to be just a totally intractable problem with the chemical copying of RNA. There's also this problem that if everything worked perfectly, and we could take a single strand of RNA and copy it and make a perfect double-stranded helix, it would be impossible to get the strands apart without enzymes. If you just heat something like that up, the duplex is so stable the strands don't come apart. So it would be a dead end. And those are probably the worst problems. But then there's another half dozen problems which also could be fatal problems. So really all of these things need to be solved if we're going to have a simple yet robust way of copying RNA sequences. Okay, so I'm not going to go over all of them. I just want to focus on one, and that is this regiospecificity problem, or the problem of making a backbone that's homogeneous. Okay, so what you're seeing over here is a diagram of the structure of a normal RNA molecule. And just showing the chain of sugar, phosphate, and the nucleobases are shown as N. So that could be A, G, C, or U. Okay, so the backbone, though, is very regular. It's always this oxygen connected to a phosphate going to this one. So this is called the 3'. You have a phosphate, the 5' oxygen, 3' to 5', 3' to 5', and so it goes for hundreds and hundreds of bases. Always 3' to 5'. The enzymes that make RNA, that make messenger RNA, ribosomal RNA, tRNAs, they never make a mistake. They always make 3', 5' linkages. But when you try to do this kind of RNA synthesis without enzymes, then that breaks down. And now what we see is we get a backbone that's messed up, because sometimes we have these 3' to 5' linkages, and sometimes it's the other oxygen that reacts. And now we have a 2' to 5' linkage. And so as you go down the chain, you have this random mixture of 3' linkages, 2', 3', 2', and the order cannot be inherited. Right? There's nothing about the sequence or the bases that gives you any information about which linkage to make. So again, this problem was recognized in the late 60s. It was felt that this is one of the biggest problems with RNA as such. How could you ever build a useful structure if the backbone had this kind of heterogeneous variation that couldn't be inherited, right? You couldn't build a nice three-dimensional folded structure that could catalyze a reaction. So this was viewed as a problem that had to be solved. And people spent a lot of effort trying to find conditions that would give you more of the right backbone structure and less of this messed up backbone structure. Okay. But the problem was never fully solved. And eventually, people more or less gave up on it and said, well, maybe we should give up on RNA as the first genetic material and think of something that doesn't have this problem. Right? There are lots of other RNA-like molecules where this is not even an issue. And so maybe one of those came first. And then RNA came along later once we had enzymes that could take care of this problem. Okay. The trouble was this view that this heterogeneity is a problem had never actually been tested. And so that's something that we, for kind of complicated reasons, decided to do recently. And the results were kind of surprising. So this is an RNA structure. This is a picture of a three-dimensional folded structure of an RNA molecule that makes a little cavity that likes to bind this molecule that is shown in the green surface. This is a metabolic cofactor called FMN. This is an RNA molecule that was evolved in the laboratory. It's not a natural RNA molecule. But it's a complicated, intricate, folded three-dimensional shape. And just the sort of thing where you'd expect that if you mess up the backbone, you're going to disturb the geometry of this structure and it shouldn't work anymore. Right? But using very simple chemistry, we can make versions of this molecule where we put in that kind of perturbed backbone. We can dope in different levels of 2' linkages and ask what happens. And the answer is, well, the more 2' linkages you put in, the worse it gets. Okay, so this is a measure of how well that molecule binds to the aptamer. If you put in 10%, it's about 3-fold worse. 25%, it's maybe 6-fold worse. If you make it completely random, 50-50, it's only about 10 or 20-fold worse. It actually still works fine. I mean, this is still pretty good binding. The molecular recognition is still there. So it kind of looks like there's something about the flexibility of RNA that this little backbone heterogeneity is maybe not that serious. Of course, we wanted to test it in a different molecule. This is an RNA molecule that's found very widely in nature in many, many organisms. It's called the hammerhead ribozyme. And it's an RNA molecule that folds up into this nice three-dimensional structure and cuts itself at a unique position. And so it's a catalytic RNA molecule, one of the simpler ones. So we decided to do the same experiment, just dope in different amounts of that backbone perturbation. And so this is what happened. We put in 10, 25, or 50% of these non-natural backbone linkages. And sure enough, the activity gets worse. 10%, it's actually not too bad. 25%, it's down significantly. 50%, we don't see anything. Okay, but still, at these intermediate ranges, which are what you get with the chemical synthesis of RNA, there's still activity. And remember, this is a ribozyme that's evolved for millions, maybe billions of years to work as something where the backbone is homogeneous. And yet it can still tolerate this substitution with the different backbone chemistry. Okay. So what do we learn from this? So first of all, the most obvious lesson is that some backbone heterogeneity is probably fine, right? And in fact, if an RNA molecule is evolving under conditions where that heterogeneity is going to be present, it's almost certain, I think, that you could find sequences that really don't care so much, that will work reasonably well with that mixed backbone. Now, the other really, really nice thing is that when you put in these non-natural, or they're non-natural today, but they're chemically natural mixed linkages, it solves one of those other fatal problems with RNA, which is that the duplex is too stable to get the strands apart. By having this mixed backbone, the melting temperature of the duplex becomes lower, and now you can just heat things up, the strands can separate, now you can copy them again, and you can go through multiple cycles of replication. Okay. So instead of being a fatal flaw, this idea that using chemistry you can't make a homogeneous backbone, instead of being a fatal problem with RNA, we now think that that might actually be the reason that RNA emerged as the first biological polymer, right? Because its heterogeneity solves this duplex stability problem. In fact, this is a little more speculative, but you could even imagine that suppose you have some useful sequence, right? You make a bunch of copies of it using this chemical synthesis. They're all going to have their 2' linkages in different positions, right? Because it's just a random process. Some of them will have those 2' linkages in positions that don't interfere with making a folded structure. So it could still be a good catalyst, right? But because it makes a stable folded structure, it'll be really hard to replicate those, be hard to copy them. Other copies, though, might have 2' linkages in a way that interferes with folding to that nice three-dimensional structure. So they'll be open, they'll be good templates. Right? So you can have your cake and eat it too. With the same sequence, because of this heterogeneity, you could have some copies that are good catalysts and other copies that are good templates. So our thinking on this problem is just completely inverted from thinking that this is a horrible problem that we have to find a way around to thinking that this is really maybe the essential and elegant feature of RNA that let it get started. Okay. So I just want to end by saying a few words about possible geological scenarios for where this kind of simple primitive cell could have actually existed and grown and divided. So this is a diagram taken from a recent Scientific American article of what a primitive cell cycle might have looked like, a cell cycle driven by fluctuations in the environment. Okay? So we think from our studies of the copying chemistry and of the membranes that these cells would be happiest if they were pretty cold most of the time, or at least, maybe room temperature down to freezing. The copying chemistry goes well, the membranes are stable. But you get to a point where all the sequences that could be copied would be. And then what we know is you have to heat them up to get the strands apart so you can go through another cycle. So a nice way of doing that would be to get caught up in a plume of hot water, this sort of thing that you see hot water streams coming up from hydrothermal vents in geothermally active areas. And then, so they'd be hot, but only briefly. So that way these delicate molecules don't get a chance to degrade. And then they get immediately cooled off again, and you can go around this cycle. Okay? So it turns out that I think this is actually a pretty reasonable kind of environmental scenario. There are still places on, many places on the modern Earth that look sort of like this, at least geophysically, if not chemically. So there are these famous lakes in the Antarctic. And so there's abundant life under the ice. So there's liquid water because of the heat rising from below. These are stromatolites, bacterial microbial assemblages growing on the side of the slope of this lake. But even more relevant, perhaps, are environments like Yellowstone Lake, which you can see here in the winter. So a large ice-covered lake, but underneath the ice are hundreds of hydrothermal vents that have been very carefully mapped. And they're basically little conical structures, and water coming from underground that's been heated essentially by the magma chamber underneath rises up, and these release plumes of hot water that range in temperature from sort of warm to really hot. Okay. So here's a picture of one of them in the summer, so you can actually see it. You can see steam rising here. And of course, all around Yellowstone, there are lots of smaller pools and geysers. It's a very active area. This is the kind of environment that we think would be necessary to support the growth and division of the kinds of primitive cells that I've been talking about. Okay. So just to conclude, I think, apart from the specifics of solving some of the individual problems and trying to build up a pathway, maybe the most important lessons that we've learned from doing all of these experiments is that there are really a lot of unexpected physical and also chemical phenomena out there just waiting to be discovered. And these are things that you can't possibly come up with just by sitting back in your armchair and thinking about the problem. Right? You really have to go into the lab and do experiments and study these molecules and look at their properties. And the second interesting lesson, I think, that's something that we've seen at least a couple of times in my lab, is that we often start off with very artificial, constrained model situations, which is what we need in order to begin studying something in a reproducible way. But once we have a proof of principle, we can work back step by step into messier, more natural scenarios. And often in doing that, that's precisely where we discover new physical phenomena that can be really relevant. And so again, this is part of the process of discovering the properties of these systems that I think eventually will let us build up a coherent pathway going all the way, hopefully, from really simple chemistry up to primitive forms of life. Okay, so I've mentioned a few of the people along the way that did some of these experiments, but over the years I've had an enormous number of incredibly brilliant, talented students and postdocs who were really the ones that did all of this work. And so thank you for listening to the story.